Hi, in this video we're going to explore how to cluster your antibodies by various criteria. So here I already have a pre-set up project where I have my samples. Actually, I have 4 samples here, and those samples are coming from 3 rounds of selection with phage displays. I already processed this data with amplicon alignment. I already redefined my clonotypes to get rid of duplicated sequences, protein sequences. And to do the clustering, I'm adding the clonotype clustering block. So this is a dedicated block which uses under the hood the well-known tool MMCQS2 to do the clustering itself. So let's add this block and explore what we can do here. So first of all, I'm selecting the data set that I'm going to analyze. Next, we are selecting the parameters of our clustering. And this is the key step, the very important step. First of all, we tell the tool how we want to cluster. Either we want to cluster the protein sequences or nucleotide sequences. In this case, and this is a common case, we're going to cluster our antibodies by their amino acid sequences. Next, we are selecting what features of our antibodies we're going to use for clustering to calculate the similarity and assign antibodies to the same cluster. Here I have a synthetic library, which is a bulk synthetic library. It's VHH, it's nanobody. So here I have all of the CDRs and frameworks or the whole VG region as an option for clustering. So for example, you can cluster just by CDR3. It will do the clustering and assign two antibodies to the same cluster if the protein sequence of CDR3 of both nanobodies met our threshold, our similarity criteria. Sometimes when you did, for example, a synthetic library with variability inside the CDRs, but the frameworks are fixed, you don't want to use frameworks as a part of your clustering criteria, and then you can use, for example, only CDRs to cluster all things together. Or in this case, let's go into use VG region amino acid sequence. So that's exactly the sequence, the protein sequence of VG region will be used to cluster antibodies together. So the next parameter is the alignment score. And there are 2 options. One option is the exact match, which will calculate the similarity, the identity of antibodies by considering the protein sequences of PDG region and calculating the number of matching amino acids divided by the total length of the sequence as the similarity score. The other option is BLOSUM, and we're going to use BLOSUM right now. This is the true alignment, which is calculated using the BLOSUM scoring matrix, which takes into account the biochemical properties of the proteins. So that's what we're going to use here. The next options are really crucial. That's minimal ID. There is some default. This default is actually good when we work with CDR3, for example. When we work with CDR3 and when we cluster by CDR3, 80% of identity, more or less reasonable default. So basically 80% of the length of the CDR3 should be identical, should match between 2 antibodies to consider them belonging to the same cluster. But if we use the whole VDG region, then 80% is very, very low identity threshold because VDG region is long, maybe 100 proteins. And given that the V region is very conservative and there are other conservative parts, 80% is too low. So we are going to use like 95. The next parameter is coverage threshold. So this is also an important parameter. When we specify the identity of 95%, there is no reason to specify the coverage threshold less than 95%, and the coverage threshold actually tells the clustering tool what is the difference in the length of the sequences that we are considering we allow. So this is a sequence-based clustering, and this is the threshold which controls the difference between the lengths of sequences. So let's run the analysis. While running, we can actually go to the logs to see like the raw logs of the alignment, of the clustering tool used under the hood. Yeah. The processing takes time, especially for the big project. It may take some memory also. So make sure, by the way, in the advanced setting, there is like the memory and CPU that you can allocate to this job. And now the analysis is complete. So here we have a table of all of the clusters that we have in the day. So let's sort by the cluster size to see whether we have some big clusters. And yeah, we have some big clusters actually, which is interesting. Actually, there is a cluster size histogram showing the general distribution of the sizes of the cluster. So the majority of our data is actually let's go to the axis and change the number of beans to some large number like 70. Yeah, so for the majority of our antibodies, they are actually singleton. The individuals don't belong to any clusters, which is very natural actually. So let's go here, back to the main. So we have the centroid sequence of the cluster that's calculated by the MMCQS. Yeah, the protein sequence of the centroid, the radius of the cluster, and the radius actually is uh, the distance from the centroid to the foremost representative of the cluster, half of that distance, well, no, that distance, the diameter is double of the radius, obviously. Yeah, and actually we see that for some smaller clusters, the radius may be larger, yeah, which is also interesting. So the next thing we can do, and easy peasy thing, we can click on the individual cluster to explore the content of this cluster on the multiple sequence alignment and see what's the general motif of this cluster. And it's a nice thing, actually, we can visualize here not only the biochemical properties of the proteins, but for example, the positions of the CDRs to see where exactly the diversity of this cluster is concentrated. There are some mutations in the CDRs. Yeah, obviously the most variability should be in the CDRs. That's what expected. And yeah, we can also see whether there is like some lineage tree structure in the cluster. And by the way, this is a multiple sequence alignment. This is a really powerful tool. We have Platforma to explore the similarity of our nucleotide and protein sequences. And the final thing is the top cluster plot. This is just for every sample. We have the biggest clusters here on the bubble plot, and the color and the size of each dot here is proportional to the size and the abundance of the cluster. So once that's done, we can, for example, use Clonotype browser to see to which cluster each of our antibody sequences belong. Like here, let's go to the overlap, just for example, and in the columns, let's see whether we have a cluster ID. We have cluster ID, we can make it visual. So all of the data is automatically integratable to each other. And here we have the cluster ID of our antibodies. That's not very interesting. The crucial part and the most important part is that this cluster information is available downstream. So for example, when we do the enrichment analysis, and for that, we have a dedicated guide. In the enrichment settings, we can select the abundance of individual antibodies, or we can select the abundance of the clusters to find, track the enriched clusters across our conditions, across our rounds of selections, etc. But this we will do in the next guide. Thank you, and stay tuned for the next nice tutorials.